Hi guys, in this video, we will learn about polysaccharides. This is a molecule of maltose. Maltose is formed via condensation of two monosaccharides. What happens if we continue the condensation process with more and more monosaccharides? This will result in the production of a long chain or a polymer of carbohydrates known as polysaccharides. An extra but essential piece of information that I would like to give out here is that in a single chain of polysaccharides that is made up from the condensation of many alpha-glucose, one monosaccharide residue is connected to the next monosaccharide residue via alpha-1,4-glycosidic bond. However, what is the name of the bond if the molecule is branched? As you can see, at a branching point, the bond is formed between carbon number 1 and carbon number 6, creating alpha-1,6-glycosidic bond. Therefore, if the polysaccharide is unbranched, there is only the presence of alpha-1,4-glycosidic bond. But if the polysaccharide is branched within a single strand, there is presence of alpha-1,4-glycosidic bond, but at each branching point, they are connected to each other via alpha-1,6-glycosidic bond. These are the characteristics of polysaccharides. Polysaccharides are not soluble in water. For example, we know that rice is full of starch, and starch is an example of polysaccharides. Does our rice dissolve when we pour soup into it? No, right? So that's why we say polysaccharides are insoluble in water. Polysaccharides are also not crystallizable, and polysaccharide is also not sweet. If we continue the condensation process of many alpha-glucose molecules, we will get a polysaccharide that is perfect for storage purposes. Why? This is because the bond angle between many alpha-1,4-glycosidic bonds will add up, resulting in a helical-shaped molecule. Look here, there's a slight angle or kink within this alpha-1,4-glycosidic bond. If the chain is made up from the condensation of many alpha-glucose molecules, the alpha-glucose molecule residue will be connected to each other via alpha-1,4-glycosidic bond. As you can see in this diagram, the bonds are all facing the same way, facing inside, 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 and inside. Since we know that there is a kink within the alpha-1,4-glycosidic bond, and since all the bonds are facing the same way, the effect of the kink on this section will be magnified by this section, by this section, by this section, so on and so forth, giving the overall effect of coiling onto that chain. Helical molecule is more compact and does not occupy much space. So that is why a polysaccharide, which is formed via condensation of many alpha-glucose molecules, are more suitable for storage. Could you imagine how much space you could save if all of your pencils and pens and rulers could be rolled up? Exactly. Secondly, polysaccharides does not dissolve in water. Therefore, it does not disturb the cell's water potential. Lastly, polysaccharides can be easily broken down to release glucose. This is an unbranched polysaccharide made up from the condensation of many alpha-glucose molecules. As you can see, it forms a helix, making it more compact, using less space, therefore very suitable for storage. This is a diagrammatic representation of glycogen. Glycogen is carbohydrate storage in animals. This is a much simpler view of glycogen. It is branched, but still it is a helical molecule. Now let's have a look at the examples of polysaccharides that are formed from the condensation of many alpha-glucose molecules, which make them suitable to act as carbohydrate storage. For animals, we know that animals store excess carbohydrate in the form of glycogen. And as you can see from the diagram, glycogen is a branched polymer. Since glycogen performs a storage function, it must be made up from the condensation of many alpha-glucose molecules. So therefore, there must be the presence of alpha-1,4-glycosidic bond. And since it is branched, the branching is formed between carbon number 1 and carbon number 6, creating alpha-1,6-glycosidic bond. Plants, however, store their excess carbohydrate in the form of starch. There are two types of starch, the unbranched starch and the branched starch. The unbranched starch is known as amylose. Since it is unbranched, there is only alpha-1,4-glycosidic bond present. The branched starch is known as amylopectin. Hence, there are two types of bonds present, alpha-1,4-glycosidic bond and alpha-1,6-glycosidic bond. It is very difficult to distinguish between amylopectin and glycogen if you are given a diagram. The only difference is that glycogen is more highly branched compared to amylopectin. If the condensation of many alpha-glucose molecules will form a carbohydrate polymer which is perfect for storage purposes, the condensation of many beta-glucose molecules will form a carbohydrate polymer which is perfect for structural purposes. The example of such molecule is cellulose, which is the building block of plant cells' cell wall. Why is this so? Here is a diagram which shows part of cellulose polymer. As you can see, one beta-glucose residue is connected to the next beta-glucose residue by beta-1,4-glycosidic bond. Although there is a bond angle or kink within beta-1,4-glycosidic bond, unlike glycogen, amylose and amylopectin, where the bonds are all facing the same way, for cellulose, the bond alternates. As you can see, this bond is facing this way, this bond is facing this way, this bond is facing this way, and so on and so forth. Therefore, although there's a bond angle within beta-1,4-glycosidic bond, but since the bonds alternate, therefore the bond angle or the kink in this section cancels out the bond angle or kink in this section. Since the bond angle cancel each other, this results in the formation of a straight chain, and thus forming a fiber. Besides using straight chain, you can also use the word linear chain. Cellulose is unbranched, therefore there is no such thing as beta-1,6-glycosidic bond. However, many different cellulose fibers can form hydrogen bond with each other in an interaction called cross-linkage. Let's take a look at this diagram. This represents one cellulose fiber. As you can see, it is connected to the next cellulose fiber via hydrogen bonding. This will then forming like a net, and the overall effect contributes to the toughness of the cell wall. Do you remember that one of the lesson objectives that you need to achieve as you're learning about carbohydrates is you need to be able to compare the structures and functions of starch, glycogen, and cellulose. So please make sure that you take some time to find the similarities and differences between these different polymers. We will definitely discuss regarding this matter in one of our classes.